Okay. 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 Hello, everyone. Thank you for coming to this YouTube channel to watch this presentation. So, my name is Matt DeMars, and I'm a postdoctoral researcher in the Department of Natural Product Biosynthesis at the Max Planck Institute for Chemical Ecology. Today, I'm going to be talking a little bit about resurrecting the past, bringing ancient proteins back to life, and studying them to see what they can teach us about evolution. So first, I just want to start kind of broadly. So biology is obviously a huge field, and it has many subdisciplines. So one, of course, is evolutionary biology, dealing with the evolution of organisms over time. And phylogenetics is a related discipline, looking at the relationships among or within groups of different organisms. A field that is more familiar to me is molecular biology and biochemistry. And so if you merge these two fields, you get sort of this interdisciplinary area, this so-called, this functional synthesis, as they call it, of these two areas, where you have molecular evolution, which is looking at the change in sequence composition of biological macromolecules, DNA, RNA, proteins over time, and also a sort of a little more niche area, which I'll be talking about today, sort of a sub discipline of that would be like paleoenzymology, where we're looking at resurrecting ancestral enzymes to study their properties. So I'll be talking about this technique of ancestral sequence reconstruction that is used in this overlap area of evolution and molecular biology. So what is ancestral sequence reconstruction? It's a sort of self-explanatory, but basically it involves reconstructing genes from ancestral organisms. So you have modern genes, you perform this ancestral sequence reconstruction, ASR as I'll abbreviate it, and you get ancestral genes through this process. And in turn, these ancestral genes can be resurrected in the lab, basically by putting them in a suitable heterologous host, like E. coli, and then expressing the corresponding proteins and studying their properties. And so in classical biochemistry, basically we have a bunch of proteins that are modern, so extant proteins with different properties, and we may study them to understand how they differ, and to see basically to study how their sequences lead to different structures and different functional properties. And so this is so basically these modern proteins all represent these tips of this phylogenetic tree that I show here. And so this classical approach or horizontal approach is, as I said, looking just at the extant proteins and comparing them. However, there's an alternative approach where we sort of look vertically at this phylogenetic tree into the past, basically, and looking at the nodes of this tree, and these nodes basically represent ancestors of all of the modern proteins that are shown here in different colors. And so what we can do is do this vertical comparison of not only the extant proteins but also the ancestral proteins to understand what changes they underwent in order to have the different functions that we observe today. So how is ASR performed? So it's a fairly straightforward, conceptually it's fairly straightforward. Basically, you start with a collection of homologous proteins from diverse species that you're interested in looking at. And then you perform a multiple sequence alignment where you align all of the different primary sequences of the protein. So these are the amino acid, primary amino acid sequences that are aligned. From there, you build a phylogenetic tree, shown here for an example. And then you can go from here, and all of this, by the way, is done computationally, or at least assisted greatly by computational methods. And from here, from the tree, you build or reconstruct the sequences that are present at the ancestral nodes, shown here as gray circles. And then once you have the ancestors reconstructed, you can then have the gene synthesized by a company, or if it's only a few mutations, you can introduce them yourself in the lab. But you can have the gene then synthesized and put into a suitable expression vector, and then have a heterologous host express the protein, and then you can go on to study the protein to understand how it's working. So first, collecting homologous protein sequences from various species. There's a number of databases available for this. The most popular ones being the NCBI, which includes a bunch of sequence data from many organisms, as well as UniProt. And UniProt is focused more on proteins and looking at, if you're just interested in looking at protein sequences directly, you can use UniProt for most cases. And so you have a bunch of sequences. Now you can perform a multiple sequence alignment. This is a very critical step in performing ancestral sequence reconstruction. There are a number of available tools. These are different softwares that are available online, or you can download them and perform them on your computer. And they just they're just different algorithms that are designed to optimize alignment of sequences with differing degrees of similarity. And so you can try, a lot of times people try multiple different alignment tools to see what gives you the best outcome. From here, once you have your good alignment, you can construct a phylogenetic tree. This is another very critical step in this process. There are several different kinds of methods that you can use. The most popular ones, and the ones that are most especially suited for ASR, are maximum likelihood. So basically, maximum likelihood has this explicit sequence evolution model incorporated into it, and these models are basically probabilistic descriptions of how the amino acid or DNA sequence changes over time. And so this ML method builds a tree that has the highest likelihood of being correct based on the alignment that you give it. So that's why also the alignment is very important to have, robust. The other popular method used is a Bayesian method. I won't talk about the details here, but it's basically using Bayesian inferences to build the tree instead of ML. The final tree that you use for ASR should be very well supported topologically. This means that basically all of the nodes have high support values, high bootstrap support values, for instance, if you're performing ML, or high posterior probabilities for if you're performing it using the Bayesian method. And it should also be consistent with any other known phylogenetic data that's available. Again, there's a number of online available tools for this, very popular ones like IQ-Tree I use. Mr. Bayes is a very high state-of-the-art Bayesian tool that you can use. Other softwares like PAML, PhiML, RaxML, these are all, again, maximum likelihood methods that are popular and very good for this. So once you have your tree, you can start to reconstruct your sequences at the ancestral nodes. Again, you can use ML or Bayesian methods for this. Sequences you can reconstruct can be up to hundreds of millions of years old. I think the record is, on the order of the age of life itself, 4 billion years. The reconstructed sequences are ranked by probability of representing one of many possible ancestral sequences. So the sequence you get is not necessarily the true ancestral sequence, but it's one of many possible ancestral sequences. And the key is that for most applications, you're going to want to sequence the gene, or construct the gene and express the corresponding protein to test its properties. And these ancestral sequences that correspond to that protein may not be identical to the true ancestor, but they will likely exhibit the same properties as the ancestral protein. And another possibility, if you're interested in doing it, is determining the age of this ancestor using a molecular clock model. But this is sort of a little finicky, and it's hard to get an exact age, so it'll always be sort of an approximation. Again, available tools for this. The most popular one, I believe, is PAML, which is just phylogenetic analysis by maximum likelihood. The one I've used is this FastML. It's a more user-friendly, online, server-based software program you can use. There are a number of other ones. For instance, this FireProt ASR is a new thing that I found that is very user-friendly and allows people with essentially no experience in performing this to do it. And it does everything basically automatically. All you have to do is feed it your query protein sequence. And so again, once you construct these sequences, most people will want to have the gene synthesized and then cloned and expressed in a suitable heterologous host. So why is ASR performed? There are a number of good reasons as to why people might want to perform this technique in their own research. So again, this is really, instead of a horizontal approach, which is classical biochemistry, this is a vertical comparison of extant and ancestral proteins. So it allows you to answer several questions you can't answer otherwise. Namely, where and when did functional shifts in protein activity occur? So by where, I'm referring to where in the protein sequence, so what mutations caused a protein's change in function or biophysical properties. And then when, so when during evolution did these mutations arise? It can also be used to answer broader questions about evolution in general, such as, does evolution proceed in small steps or in large leaps? Is evolution reversible? And how does complexity evolve? These kinds of broader questions are also approachable using this technique. Another thing that I won't talk about after this is a number of industrial applications of ASR, mostly in the biotechnology and biomedicine fields. So it turns out that that ancestral proteins are often exhibit high thermostability and broader substrate promiscuity if they're catalyzing an enzymatic reaction. And so these are very useful traits for an enzyme to have for biotechnological application. And biomedicine, you can also use ASR to reconstruct ancestral proteins and genes from viruses, for instance. And so you can look at how a virus has evolved over time, and it can be used potentially to develop more effective vaccines. So let me talk about a little bit about a few examples of ASR having been used in different labs to understand evolution. And I'll focus today on a couple of studies that I found interesting, having to do with myoglobin and hemoglobin. These are oxygen transport, storage and transport proteins that are found in invertebrates. So first I'll talk about this interesting study from Science back in 2013, which traced the evolution of mammalian diving capacity by looking at net surface charge, the change in net surface charge in myoglobin over time. So myoglobin is shown here on the left. It was the first protein whose structure was elucidated back in the late 50s, actually. It's responsible for binding and storing oxygen in muscle tissue. And notably, high levels in muscle allow aquatic mammals like these sea lions or these whales here to hold their breath underwater for long periods of time, up to more than 1 hour. So basically, what they wanted to answer in this study was, how did aquatic and semi-aquatic mammals acquire the ability to hold their breath for extended periods underwater? So it was already known that it had to do with high levels of hemoglobin in their muscle tissue, but they wanted they wanted to understand what about their hemoglobin made it possible for it to be in such high concentrations in these tissues. So they modeled the evolutionary history of myoglobin over about 200 million years using ancestral sequence reconstruction. And they found that there's this adaptive molecular signature of elevated myoglobin net surface charge, so the overall charge on its surface, in lineages of diving aquatic mammals. And so basically, higher myoglobin net surface charge leads to increased intermolecular electrostatic repulsion, as they hypothesized. And this in turn leads to the ability of myoglobin to accumulate at higher levels in muscle tissues without aggregating. And this in turn leads to the longer dive times that are observed in these mammals. And so ultimately, they were able to reconstruct the evolution of diving capacity across major groups of mammalian divers. You can see here the ancestors of modern whales, for instance. This Basilosaurus had elevated myoglobin surface charge, and so this was correlated again with higher concentrations in the muscle tissue and higher diving times. So you can really look back. This technique really allows you to look back not only at the proteins and how the proteins have evolved, but can also be used to make inferences about the animals to which those proteins belonged. And so that's really cool, I think. Another related study looked at, this is a more recent study in Nature from last year, and this study looked at the origin of complexity in hemoglobin evolution. So hemoglobin is similar to myoglobin, but it's a little bit more complex in terms of its structure and its function. So unlike myoglobin, hemoglobin is responsible for transporting oxygen throughout the body. So it binds oxygen in the lungs or gills in aquatic animals and releases it in the periphery. And it's able to do this because oxygen binding and release is cooperative. So basically, there are these 4 subunits. It's a hetero tetramer made up of 4 subunits, 2 alpha chains and 2 beta chains, and these are homologous proteins, each of these chains. So it's basically 4 separate proteins in one large quaternary structure that makes up the total molecule. And so once oxygen binds to one of these chains, the affinity for oxygen for the other chains increases. So it allows oxygen to bind more quickly and to be released more quickly in the periphery. And another reason that it can be bound and released so readily is that its oxygen affinity is lower than the affinity that myoglobin has for oxygen. So the question is, how did hemoglobin evolve? Again, so we have this heterotetramer of alpha beta subunits. So how did that evolve from presumably this monomer that is similar to myoglobin? So the researchers inferred the phylogeny of hemoglobin and related globins using collecting a bunch of different sequences from different organisms and performing an alignment, as I've talked about, and reconstructing this tree. They ended up reconstructing 4 different ancestral globins, the ancestor of the alpha subunit, the ancestor of the beta subunit, and the ancestor of both of those. And these are from, so the ancestor here existed greater than 400 million years ago, before the common ancestor of the jawed vertebrates. So this ancestor, let's see here. Ancestor alpha/beta here, they found by expressing it and testing its biophysical properties, they found that it's a homodimer instead of a homo or heterotetramer, and it binds O2 with high affinity non-cooperatively. However, if you introduce just two mutations into this ancestor at one of the interfaces, now you acquire this ability to to form a homotetramer. And if you introduce just 2 to 4 mutations at each of 2 interfaces, now you start to get this heterotetramerization that you see in modern hemoglobin. And so they found ultimately that cooperative O2 binding arose with the ability to tetramerate. I can't say this word, tetramerize. And so basically, you start from this ancestor of myoglobin and hemoglobin, which is a monomer. Then you go to this ancestor of the alpha and beta subunits, which is a homodimer. So it's also shown here schematically. You can introduce only a few two mutations into this homodimer, and now you get heterotetramerization, which exhibits cooperative binding properties to oxygen, and also lower affinity to oxygen. And so ultimately, what they found was that you just need a few mutations to acquire a structural and functional complexity during evolution. So a lot of times people think about, in terms of at least protein structure, when you have a complicated protein structure which performs a complicated function, people think that you need a bunch of mutations to accumulate over time, and that it's a very complicated process. And so it's really hard to understand how this happens. But it turns out that, at least in this case, and in several other cases that this particular group has studied, you only need just a relatively few number of mutations in order to gain this complexity in structure and function. And so that's pretty interesting, and it tells you something more broadly about evolution in general. So I'll talk a little bit about how I'm applying ASR to my own research. So I work on indole alkaloids from medicinal plants, and so probably the most famous alkaloid that I work on, or that my work is related to, is vinblastine, which is an anti-cancer drug that is used in the clinic, and it comes from this plant, Catharanthus roseus. So vinblastine is has a very complicated molecular structure, as you can see. But it's made up of basically these two different units. This one unit is shown in red here, is derived from the metabolite catharanthine, and this other unit here is derived from tibercinine. So these are two different indole alkaloids that this plant produces, and that ultimately lead to the production of vinblastine. So catharanthine and tibercinine are actually formed enzymatically from a common intermediate here, referred to as dehydrocicodine. And so this common intermediate is used by homologous two different enzymes. One is this catharanthine synthase, which makes catharanthine directly from this intermediate, and the other is tibercinine synthase, which does the same thing. But as you can see here, the structures of these two compounds are quite different. And so the question is, how do these related proteins control the selectivity of cyclization of this intermediate? There's actually a third cyclase that we've found as well that is responsible for performing this other cyclization, which forms a product that's related in structure to catharanthine, but it has the opposite enantiomer configuration. So the configuration of bonds is somewhat different. And so all of these enzymes are highly related. They're 75 to 80% identical by sequence, and they all are part of this alpha beta hydrolase superfamily. And so the question I have in my research is, how did these different cyclase functions evolve? And how did ultimately cyclase function evolve from carboxylesterase function? So these alpha beta hydrolases are usually acting as carboxylesterases. So they're cleaving ester bonds and forming basically an acid and an alcohol, which is a function that's radically different from the cyclization activity that they're performing here. So basically what I did was collect a bunch of different sequences from different species of plants and homologs of these different cyclases, and made a phylogenetic tree. This is just a small version shown here. And then I wanted to test a subset of these extant cyclase homologs. And so down here, this grouping down here are all of the cyclases. So these enzymes that are shown here at the nodes are performing this cyclization reaction. This grouping here is is unknown. So the proteins I haven't been able to determine a real function for. They're catalyzing neither cyclization nor really effective carboxyl esterase activity. However, when you go further back in time, or not back in time, but further up the tree in this case, looking at more distantly related extant proteins, these are performing ester hydrolysis on model substrates that I use. So what I've done in my research is to reconstruct key ancestral nodes and test for cyclase and esterase activity. So I've reconstructed the ancestors at the nodes shown that are highlighted by the arrows here. And I found some pretty interesting things, such as what the functions of these ancestors are here, whether they're performing, which types of cyclization reaction they're performing, as well as whether or not these nodes out here are exhibiting esterase activity or cyclase activity. But I'm not going to disclose the results. You'll have to wait till the paper comes out. It's a little bit confidential, but the work is going well, and the results have been very interesting. So yeah. So with that, I'd like to thank you for listening, and I can answer any questions if there are any. Thanks also, I should say, to the Max Planck Institute for Chemical Ecology and to the Department of Natural Product Biosynthesis, the director and Director Sarah O'Connor, for the opportunity to work on this interesting research topic.
Yes, there was actually one question that was even a little bit discussed in the chat. It's how would you answer the question if evolution works rather in small steps or in big leaps?
Okay, that's a great question. So what you can do with ancestral sequence reconstruction is look at the protein level, how proteins have evolved, to see how many changes were required to go from an ancestral function to a modern function. And so in the example of the hemoglobin evolution, the researchers showed that only a few mutations were required to go from a relatively simple structure and function to a more complex function that hemoglobin has. So in that case, at least, evolution proceeded in small steps. And so you can look at how many changes have occurred in a protein over time and see how much it affects its function. And if only a few mutations are required to really affect a drastic change in the function, then we can say for that particular case that evolution proceeded in a small step. As far as looking at more broadly, maybe at organismal evolution, it's a little bit more difficult because organisms are much more complicated systems made up of thousands of proteins and interacting with each other. But in as much as we can make inferences about the organism itself from the data that we acquire from reconstructing its ancestral proteins, the example that comes to mind, of course, is the myoglobin, the example of the myoglobin evolution, where we looked at, or the researchers looked at diving capacity over time. We're able to infer diving capacity over time by looking at how myoglobin had changed. These techniques can sort of indirectly give you some insight into how much change was required to go from the ancestral state to the modern state. And you can actually get a good idea as to how many changes were required, and was this really a big leap forward in evolution, or was it just a very minor step, relatively speaking? And so, yeah, it can in this way, basically, looking at the sequences and of the proteins that you can reconstruct, and using that data to extrapolate to larger systems, including the whole organism, you can make these sorts of inferences.